Cell metabolism offers a window into cell function, revealing key drivers of growth and division, stress response, and survival. But studying metabolism doesn't need to be complicated. Take a streamlined approach with Promega metabolite assays, designed to help you explore cellular metabolism easily, efficiently, and with confidence. Let's take it one step at a time. Just getting started with metabolism research, it begins with asking the right question. Understanding cell metabolism can offer insight into everything from cancer progression to immune cell function, and with the right tools, it's easier to ask better questions. You might be asking, are my cells shifting from glycolysis to oxidative phosphorylation? Or how does nutrient availability affect cell stress and survival? Whatever your question, the first step is measuring how cells are using and releasing key metabolites. Metabolic activity can be measured from a variety of biological sources. Promega metabolite assays are compatible with multiple sample types, including but not limited to cell culture media, cell lysates, serum, and tissue homogenates. These sample formats integrate easily into existing experiments, so you can collect meaningful data without reinventing your workflow. Once you've chosen your samples, collection and preparation are simple. Most Promega metabolite assays are endpoint assays, but many are also well-suited for time course experiments. If the metabolite is secreted into the culture media, you can sample at multiple time points without disturbing the cells. No centrifugation or extraction necessary, just pipette and go. For intracellular targets, a brief lysis step is typically all that's needed. When it's time to run your assay, the protocol for Promega metabolite assays is designed for ease and consistency. Most follow a simple format. Add sample and reagent, mix gently, then read the signal on a plate reader. Many of these assays use bioluminescent chemistry, a glow-based readout with high sensitivity and low background. This makes it easier to detect subtle changes in metabolite levels, even in complex samples. Data from Promega metabolite assays can stand on their own or support broader biological insights. Results are typically available in under an hour and can reveal shifts in glucose uptake, lactate secretion, redox balance, amino acid metabolism, lipid use, and more. Use these results as standalone data or pair them with transcriptomic, proteomic, or metabolomic methods to verify pathway activity and gain additional context. Every experiment is different, and your workflow should adapt to your needs. Whether you're studying a single metabolic process or mapping a broader network, Promega metabolite assays are modular, scalable, and compatible with your existing tools. You can start small or scale up, mix and match assays, and build out your research over time. It's a flexible platform designed to support researchers at any level.